Okay, with lab 2, we're going to look at cell structure as well as the process of diffusion and osmosis, moving material into and out of the cell. With the cell, we have a cell membrane. This is called the plasma membrane that surrounds the whole cell. It is a bilayer of phospholipids, and it is going to regulate what enters and exits the cell as well as the internal environment versus the external environment. In the very center, we have the nucleus. The nucleus is the brains of the cell. It is going to house our DNA, which we call chromosomes. In this model, though, the DNA is stretched out in the relaxed phase, and when the chromosomes are relaxed and stretched out, it is called chromatin. They spend the majority of their life in the chromatin phase and only condense into chromosomes when they are getting ready to divide. Surrounding the nucleus is a nuclear envelope. It's a membrane that has little holes called nuclear pores to let small materials enter and exit the cell. The DNA cannot get out, but messenger RNA can when it copies the DNA. Also found inside the nucleus is another organelle, its own organelle. It's called a nucleolus. A nucleolus, these are small transient organelles. There's usually 2 to 4 of them within the nucleus. And in general, their function is to produce the ribosomes, which are going to then exit and be involved with protein synthesis. Outside the cell on this model, we have extending from the nuclear envelope another membranous material, and this is called the endoplasmic reticulum. The endoplasmic reticulum is a channel which is going to be made of house many enzymes and will modify proteins as they move through the channels. There are 2 types of endoplasmic reticulum. On this model, we have mainly rough. It is rough because it has associated with these little white dots, which are the ribosomes. The ribosomes are going to read the information the messenger RNA brings out from the DNA, and they will make the protein, and then the protein that they produce will go into the endoplasmic reticulum, the rough endoplasmic reticulum, and be modified. Any protein that is produced and modified in the rough endoplasmic reticulum is going to be either used in the cell membrane itself or be exported out of the cell. We don't really have too much smooth, maybe a little bit here, not really. Too much smooth endoplasmic reticulum. It is called smooth because it does not have any ribosomes associated with it, and often the protein will move from the rough endoplasmic reticulum after it's been modified over to the smooth, where it will actually further modify it and produce carbohydrates and lipids, as well as having a variety of different specialized jobs in different cells. Once the proteins are produced, and they are going to be packaged up by the Golgi apparatus, and the Golgi apparatus will then pinch off little vesicles which will go to the plasma membrane and will shoot out their material through exocytosis, where it just kind of spits it out of the cell. And also in our cell membrane, we have the mitochondria. These are the powerhouses. They are going to make the ATP in cellular respiration. These are mitochondria. These are double membraned organelles. This is also a cross section. These are without it being cut, just so you know that all of the orange here is the mitochondria. We also have the centrioles. The centrioles are located in the zone called the centrosome, the brown area. That's their space where they live. And your centrioles are going to be involved in cell division, which we will talk about in the next lab. We have lysosomes. Lysosomes are organelles that are filled up, they're vacuoles, that are filled with digestive enzymes. You can think of this as the cleanup crew for the cell. And they are going to go around and engulf old organelles. Large particles, such as an invading bacteria into the cell, will be engulfed by the lysosomes, and then they will digest it, and they will then package up any waste product, and it will go to the cell plasma membrane and will be spit out through exocytosis. We have another set of vacuoles called perioxysomes. That's these gray or blackish looking ones. The perioxysomes are filled up with digestive enzymes specifically to break down fatty acids, and a second set of enzymes to break down the byproduct of fatty acid digestion, which is hydrogen peroxide. That is toxic to the cells, so we need to remove that also from the cell. Okay. Not seen on this cell model are extensions that can occur outside from the plasma membrane. In some of our cells, we will have these little long finger-like extensions that will come right off the cell membrane, and these are called cilia. And the finger-like extensions are going to wave back and forth, and they're going to be used for movement, to move material by the cells, or if it's a little single-cell organism, to move material to their mouth. We also have microvilli. Microvilli are going to be more stubby on the outside. And again, these aren't on every cell, these are just on certain specialized cells. Microvilli are going to be found in your digestive system, and especially in the small intestine, where they're used for absorption of nutrients. And then we will have a flagella on one cell in the human body, and that is the sperm cell. And that is a long tail that is used like a whip so that the sperm can swim.